I'm Jim Baxter, and this is a lecture on elements in ecosystems for general ecology. I chose this spectacular picture of a waterfall cascading over this cliff because it highlights a fundamental concept in ecosystem ecology. That is, that materials move between biotic communities and the abiotic environments in which they live. The movements of these materials in ecosystems is affected by chemical, physical, and biological processes that are the underlying mechanisms that drive their movement. To understand the movement of these elements, of which there are many, it's helpful to think of the Earth system in terms of different compartments or pools in which these different elements reside, and the different processes that are responsible for moving an element from one pool to another. These are the learning objectives for this lecture, and as I've been doing, I'll let you take a look at these on your own. Let's move on. On Earth, all matter cycles, and it cycles fundamentally through different compartments. Those compartments are the atmosphere, the hydrosphere, the lithosphere, and the biosphere. You're probably familiar with some of these, maybe not all of them. The atmosphere, of course, we've talked about that. The hydrosphere is all of the water system of the entire Earth, including oceans, lakes, streams, rivers, groundwater, and so forth. The lithosphere is the geologic component of the Earth, including, of course, its core, the crust, all of the rock minerals, plate tectonics that are made of, rocks and so forth. This is all lithospheric material. And of course, not least of which is also the biosphere, of which we're a component. That is the biologic component, both living and dead. The Earth is essentially a closed system with regard to all of this matter. The gases in the atmosphere, the water in the hydrosphere, the rocks and the elements and minerals in the lithosphere, and all of the biological organisms and all of the elements contained therein. It's a closed system, and what that means is that no new materials are coming into the system nor leaving it, at least not in any appreciable way. Sure, meteorites and cosmic dust and so forth find their way to Earth, and there's a very small amount of loss of materials to space as well, but these amounts are very, very small. So for all practical purposes, all the matter that exists on Earth is all the matter there is. That is, that there's a finite amount of matter on Earth actually has some important implications for how we understand the cycling of elements among these compartments and the impacts of changes in these cycles. I'll come back to this point in a moment, but before we go on, I want to introduce this term that I used in the title of this presentation, and that is biogeochemical cycle. A biogeochemical cycle involves the movement of biologically important nutrients among these different compartments of the Earth system. The term biogeochemical is used because the elements that move around among these different major compartments on Earth, they move through various different mechanisms or processes that involve both biological organisms, hence the prefix there, bio. They move through geological materials, hence the geo in the name, and then chemistry plays an enormous role as well in terms of transforming materials from one form to another. Of course, a lot of chemistry goes on inside of biological organisms and geological materials, and so these cycles are referred to as biogeochemical cycles. So organisms contain a large amount of hydrogen, oxygen, and carbon. Of course, these are major constituents of organisms. shown here, CH and O. These are constituents of carbohydrates, of course, but also proteins, DNA, lipids, etc. However, as key components of these biomolecules, they also need organisms also need nitrogen, phosphorus, and sulfur in appreciable amounts. And so these 6 elements generally are considered kind of the 6 major elements in biological organisms. So these are the major constituents. Of course, organisms also need a variety of other elements, but in smaller amounts. And these include elements like potassium, nitrogen, calcium. I guess you can read, magnesium, iron, and so forth. Although these are not required in as high a concentrations, these minor elements are certainly very, very important constituents of organisms. They're components of enzymes. They regulate the cell osmotic environment, for example. Sodium-potassium pumps are incredibly important. Calcium in bones and teeth. It's also involved in blood clotting. Magnesium is at the active site of chlorophyll, and we've talked about that, and of course how important chlorophyll is as a molecule that harvests light energy to drive photosynthesis. Iron in hemoglobin, fluoride in tooth enamel, chlorine in digestive juices, and so forth. Okay, so all of these elements are important. They're just they constitute different. They comprise different amounts in organisms. Because these elements reside temporarily in the tissues of living and dead organisms, and biological processes play an important role in how these elements move around in ecosystems, biological organisms play a really important role in biogeochemical cycles. In this lecture, I'll introduce the ways in which biologically essential elements, like the ones here, cycle. And in the next lecture, I'll focus specifically, though, on the cycling of 2 very, very important nutrient elements, carbon and nitrogen. But let's start. I'd like to start with kind of an example that I think you're all very familiar with in terms of a cycle, the water cycle. This is perhaps the most simple biogeochemical cycle, and one that you're probably most familiar with. Although it might sort of appear kind of simple in a lot of ways, this diagram is not too complex, the water cycle is tremendously important. It's important because it's really fundamentally important for moving many other kinds of elements around. When ecologists study biogeochemical cycles, they conceptualize them in terms of different compartments, and that's shown here in this water cycle, where elements reside for a period of time but can move between compartments. So compartments act as temporary reservoirs, you might say, for elements, and can receive elements as inputs and lose them as outputs and so forth. The mechanisms by which elements move from one compartment to another vary a lot in terms of the physical and chemical processes that are important in driving the transformation or movement of an element from one compartment to the other. Biological processes are also tremendously important. So, for example, the water cycle that I'm using here to just kind of introduce this concept of a biogeochemical cycle. The water cycle shown here, which of course the molecule is H2O, and what we're really fundamentally talking about, though we're talking about the movement of water, we're really fundamentally talking about the cycling of 2 elements, hydrogen and oxygen. Water moves from one compartment to another by various processes, as shown here. And when we talk about the movement of water through ecosystems in this way, and in the atmosphere, this is known as the hydrologic cycle. So it's just another name for the water cycle. The key mechanisms here that drive the hydrologic cycle are precipitation, of course, rainfall, snow, and so forth, evaporation off of land and sea, surfaces, and transpiration, of course, that is the loss of water off of plant surfaces through the stomata. In the water cycle, of course, the largest pool of water is made up of the oceans, which contain approximately 97% of all the water on planet Earth. Okay, so the key processes, as I mentioned, being evaporation, precipitation, and transpiration, but we've also got runoff and other kinds of processes going on. The hydrologic cycle is really fundamentally important, as I mentioned, because it's responsible for moving many of the elements in biogeochemical cycles through different ecosystems or through different compartments. Water is also really important, in many chemical transformations of materials, as I'm sure you're aware, that occur in living organisms, but also in the non-living components of ecosystems. And of course, as we discussed at the beginning of the semester, it's fundamentally important in determining global climate, right? In particular, patterns of precipitation. As I mentioned in one of the previous slides, Earth is a closed system. And so I want to come back to this because as such, water, as it cycles on Earth, it cycles on Earth with no net loss or gain over the long term. As a result, any change in one part of the water cycle influences the other parts. And in terms of climate change, a warmer climate, as we have discussed, due to increases in carbon emissions into the atmosphere, means more evaporation off the land and sea, right? Warmer climates, more evaporation, more moisture, more energy. An increase in this evaporation, but also in transpiration, will cause more water to enter the atmosphere, more water to move through the hydrologic cycle in general more quickly, potentially leading to increasing intensity of rain and snowstorms in various parts of the world. So all of these sort of mechanisms and interconnections through these biogeochemical cycles interact with one another in important ways. So the water cycle, in many ways, is a fundamental driver behind how other biogeochemical cycles function on Earth, and it's a good introduction to thinking about kind of how we think about biogeochemical cycles in general. So let's take a look at how a biogeochemical cycle is modeled, or kind of how we conceptualize it, and how these models can help us understand how nutrient elements cycle among reservoirs. So one of the terms that I'll use a lot, and I'll kind of use two terms interchangeably, reservoirs and pools. I mean pretty much essentially the same thing. A reservoir is simply a storage form of a nutritionally important element. Okay, so like in the water cycle, the ocean compartment, the biological organism compartment, the atmosphere compartment, those are all reservoirs for nutritionally important elements in the form of oxygen and hydrogen that make up the water molecule. Okay, and so those are shown here as boxes, and when we conceptually model a biogeochemical cycle, the box size, generally speaking, is proportional to the reservoir size. Relatively speaking, there is an enormous range in sizes of different reservoirs in biogeochemical cycles, so they aren't necessarily exact scale to scale, but larger reservoirs generally are depicted as larger boxes, and so forth. The size of a reservoir is often given as well inside the box, and depending upon the particular element that for the biogeochemical cycle that's being focused on, those numbers can range quite a bit. And we'll be talking about the carbon cycle and nitrogen cycle, and I'll be giving you reservoir sizes of those. And we'll also be talking a little bit about how we determine residence times and turnover rates of nutrients in element in reservoirs as well in a moment. So elements cycle faster through smaller reservoirs, and I'm hoping this should be relatively intuitive, but as a way of explaining that, imagine that, and if we turn to water as an example, and imagine that we have two reservoirs of water, okay, and in those two reservoirs we are going to keep track of an individual water molecule and how fast it cycles through the reservoir. Let's imagine that one of our reservoirs is like a cup of water, a coffee cup sized cup, and the other reservoir is a swimming pool. Which reservoir is it that in which reservoir will a single water molecule spend less time if we have fluxes of water moving through? So for example, if we have a little tap running into our cup, our coffee cup, and it fills the cup and that water begins to spill out of the cup, if the water is moving into the cup as fast as it is moving out of the cup, and we have the same flux rate in a pool, just simply due to the size of the reservoir, a water molecule will spend much less time in the smaller reservoir, given the same flux rate as a larger reservoir like the size of a pool. It will take a water molecule much longer time to, once it's in the pool, to leave the pool, given equivalent flux rates between those two reservoirs. The same is true for reservoirs in biogeochemical cycles. The larger the reservoir, the longer the residence time of an element in that pool. The faster it will cycle through smaller reservoirs. In our biogeochemical conceptual model here, we model the movement of elements from one reservoir to another with arrows, and those arrows are referred to as flux rates, and those show the rates of movement of elements between the reservoirs, both into the reservoir and out of the reservoir. So one of the important things about understanding a biogeochemical cycle and the way these work, because again, all matter is, there's a finite amount of matter on planet Earth, all of these flux rates basically need to balance out. If they don't, it means that a reservoir is either increasing or decreasing in size, and we'll be talking a little bit about that again when we talk about the carbon cycle, because of course we're adding carbon to the atmosphere, and so that reservoir is actually increasing in terms of the amount of carbon that's being added to it, if we consider the carbon carbon element specifically. So so we can understand a lot about how a biogeochemical cycle works when we understand the size of a reservoir and its flux rates. We can determine how fast elements are moving from one reservoir to another. We can determine the residence time of an element in a reservoir, and we can determine if the flux rates don't balance out, whether or not a reservoir is increasing or decreasing in size. And I mentioned again the enormous amounts of carbon that we're adding to the atmosphere. And the atmosphere itself is an important reservoir for carbon, and it plays a particularly important role, as I mentioned earlier, in Earth's climate. So using models like this to understand how carbon moves around, how the carbon cycle works, can help us better understand ways in which changes to one reservoir can affect the dynamics of other reservoirs for that particular element. To model the way a biogeochemical cycle works, we need to know how long an element resides in each reservoir and how fast it moves from one reservoir to another. So let's take a look at this. So let's examine a portion of the carbon cycle to illustrate this, and how we determine what's referred to as turnover rate and residence time of an element in any particular reservoir in a biogeochemical cycle. Okay, so turnover rate is the rate at which, on average, an element turns over in a reservoir, and residence time is how long that element spends in that pool. In order to be able to calculate these, and I'm going to the symbol for turnover rate will be a T and an R for residence time. What we need to know to be able to calculate this is the pool size and the flux rates out of the pool. So let's take a look at a portion of the carbon cycle. So a portion of the carbon cycle is shown here. And on the top is the atmospheric pool. That contains, and the units here are in petagrams, and a petagram is 1 times 10 to the 15 grams. Okay, so that's a lot of grams. The atmospheric pool contains 720 petagrams of carbon. If you look at, and then we have the plant and soil reservoir and the oceans reservoir, and those the sizes of those reservoirs are shown there. The arrows show the flux rates of carbon moving between these pools. So between the oceans and the atmosphere, we see that 90 petagrams of carbon per year are moving from the ocean reservoir to the atmospheric reservoir, and from the atmospheric to the ocean, the same amount, 90. And 120, there's 120 flux rate from plants and soil to the atmosphere, and that has to do with, we'll be talking about the carbon cycle later, so I don't want to go into too much detail now about how that flux rate is happening. But much of that we're referring to here is carbon moving into the atmosphere due to respiration. And of course, carbon moving out of the atmosphere into plants primarily is due to photosynthesis. So those flux rates are equivalent at 120. Okay, so the total flow of carbon into the atmosphere would be 210 petagrams per year, because if you look at the two arrows moving into the atmosphere, we have 90, that's the flux rate number 2 from oceans to atmosphere, plus the flux rate number 1, which is 120 moving from plants and soil into the atmosphere. So if we add those two together, we get 210 petagrams. Okay, so this is the input into the atmosphere, and then the total flow of carbon out of the atmosphere is equivalent at 210 petagrams per year. And so the fluxes into and out of pools really have to balance. Okay, so let's look at how we calculate turnover rate and residence time from the pool size and flux rates, and we'll focus on the atmospheric pool. Okay, so let's determine turnover rate and residence time for the atmospheric pool. The equation for that, for turnover rate, is that we take the flux rates out, and that is the sum of the flux rates going out of the atmospheric pool. And if you recall, that's going to be 120, that's F3, plus F4, which is 90, again totaling 120. Okay, so the flux rate out is 210, and the total size of the pool, P, is 720. And if we do that division, we get 0.2916 carbon, petagrams of carbon per year. Okay, so this is the rate at which carbon is turning over in this pool. Now, how long does carbon reside in the atmospheric pool? Well, that can be calculated in residence time. Okay. Sorry, the F out I just put up on the screen there that we just talked about. Okay, how do we calculate residence time? Well, it's simply the flip of the equation for turnover rate. So it's the pool size divided by the flux, the total flux out of the atmospheric pool. And of course, this would be 720 divided by 210. And if you do that division, you get 3.43 years. In other words, it takes about 3.5 years for a carbon molecule, or a carbon molecule resides in the atmospheric pool for about 3.5 years or so on average. Okay, so using these equations, we can calculate for any pool. We can calculate it as long as we know the flux rates out, the total flux out of that pool. We can calculate turnover rate and residence time for that pool. Okay. So I want to shift attention now to kind of zero in on an kind of what is generally referred to as internal cycling. Okay, elements cycle broadly across these four major compartments that I introduced to you at the beginning of the lecture: the atmospheric, the hydrologic, the the hydrosphere, the biosphere, and the lithosphere, right? But elements also cycle within a system. So if you're standing in a forest, of course there's going to be movement of elements within that system as well. There are going to be inputs of nutrient elements from the atmosphere, let's say, or maybe inputs from weathering of rocks and minerals. But and those can be inputs, and there are outputs as well, losses out of the system. Okay, but within any individual, if you're standing within any individual ecosystem, you can measure movements of elements within the system. For example, in a forest ecosystem like is shown here, where we just talked about primary productivity, right? So where plants are producing a lot of biomass, the production of that biomass, all that biomass has an enormous amount of nutrient elements in it that will cycle internally in this system through, for example, litter fall, as shown here on the right arrow, as leaves die or fall, let's say in the fall when plants lose their leaves, or just simply, due to, branches breaking or animals dying and fur and other tissues of organisms being deposited on the forest floor, that organic matter enters into the decomposer, the detrital food web, as we talked about last time, and enters and when it enters into that detrital food web, it becomes decomposed and ultimately broken down to a point where those organically bound molecules become mineralized. That is, released as inorganic forms that can be used again by microorganisms or plants taking them up through their roots or so forth. And so this is an internal cycling kind of phenomenon. So it involves the breakdown of dead organic matter and the return of the nutrients to the soils, or perhaps they enter sediments. Plants can uptake those nutrients and assimilate them into their biomass. And so there's this internal cycling. Okay. What I want to zero in on is this process of decomposition. Decomposition is really a fundamental element of this internal cycling phenomenon, because it's really what is recycling the nutrients back into the system such that living organisms can use those nutrient elements that have been recycled. So let's take a look at this process a little bit closer. So decomposition is a fundamental process, and it involves the breakdown of chemical bonds that were originally formed during the construction of plant and animal tissues, right? The construction of carbohydrates, for example, in the process of photosynthesis that we've talked about. So whereas photosynthesis involves kind of the incorporation of solar energy into organic living compounds, decomposition involves the release of that energy by breaking down these bonds and converting organic molecules into inorganic ones. And of course, because this is a biotic process, biological organisms are the ones involved in doing this. This involves the release of CO2 due to their respiration. So decomposition is a pretty complex process. It involves many different processes, in fact, including fragmentation of the organic matter, changes in physical chemical structure, and so forth. And it's accomplished by a variety of decomposer organisms. And this little diagram here just kind of shows you in an illustrative way kind of all the different kinds of organisms that are involved in decomposition. Some of you in your groups that are studying soil arthropods, of course, have learned a little bit about this and understand kind of the role of different organisms in terms of breaking down litter and decomposing it. Soil arthropods are fundamentally important, but also, of course, fungi and bacteria play an important role as well. Fungi tend to be more important decomposers that decompose plant materials, whereas bacteria tend to be the decomposers of animal tissue. So let's look at this process of decomposition. It fundamentally involves kind of the fate of dead organic matter, and I'm going to illustrate that here by this maple leaf. And so let's imagine that this leaf it's the fall. Obviously, the leaves turned color. It's fallen off the tree, and it hits the forest floor. And actually, even before it hits the forest floor, there's some decomposition that has gone on in this leaf. First of all, the tree has retranslocated some nutrients back into it, which is mainly this involves the process of breaking down chlorophyll. And in northeast deciduous forests, where you see these beautiful fall colors, that has to do with the plants retranslocating nutrients out of the leaves as they break down that chlorophyll, and some other pigments and high nitrogen molecules as well. What it leaves behind are some various pigments and so forth that take on these red, yellow, and orange colors. Before the leaf even hits the ground, though, it has fungal organisms have colonized it, and there's even some kind of decomposition or breakdown of that leaf material even before it hits the forest floor. But the main decomposition happens once it hits the forest floor, where a wide variety of organisms are that begin to break it down. So this first phase of decomposition is what's called fragmentation. Fragmentation happens when you have many different soil organisms, namely like soil arthropods, maybe worms and nematodes and other kinds of organisms, starting to chew on this dead organic matter as a source of food, right? And so in the process of that happening, they fragment that organic material. This fragmentation process is really quite important, because what it does is it actually increases the surface area that's available for colonization by fungi. And if you know a little bit about fungal anatomy, you know that fungi form quite a complex sort of a network of hyphae that release enzymes into the surrounding that are responsible for breaking down various organic molecules. They produce enzymes such as cellulases, proteinases, nitrogenases, phosphatases and so forth. All of these enzymes are responsible for breaking down this organic material further. The next step, one of the other major steps in decomposition, is mixing. There's a lot of movement of organisms in this soil system. They are fragmenting and breaking down these organic matter ever further, and as they move around in the soil system up and down in the soil horizon, they are mixing this organic material into it, and also mixing some mineral material as well. Although on the surface of the forest floor, it's primarily organic material, right? So we're talking about the O layer here, where we get ever more finely decomposed material that's being mixed up and down into the surface layers of the soil. Another fundamental process is leaching. Of course, as water enters the system through rainfall or snowmelt, as we discussed, this is an important phenomenon in formation of soil horizons. It's also important here because this leaching is responsible for some additional decomposition as well, sort of the weathering of this organic material, if you want to refer to it that way. And of course, leaching of inorganic materials that have been released facilitates its availability, for uptake by microorganisms and plant roots and so forth. The last kind of major kind of phenomenon that occurs in the decomposition process, and this is kind of the end point, is what's called mineralization. And there's actually a balance between mineralization and immobilization. The difference here is that mineralization is the release of inorganic forms of elements as a consequence of the decomposition process. Okay, so that's where the minerals, the elements have been broken down from their organic form into a mineral form that can be available for uptake. Immobilization is the process by which the decomposers actually are taking up nutrients, because of course they're biological organisms as well, and they're taking up nitrogen and phosphorus and other elements that are available in the system. And so there's a balance between these two processes in the decomposition when decomposition is occurring, such that there's mineralization of minerals being released and becoming available, but also those minerals are being immobilized by the decomposers themselves as decomposition occurs. So there's a dynamic balance between these two processes. When we talk about decomposition, we see that across different systems, decomposition rates can differ quite a bit. They differ between terrestrial systems and aquatic. They also differ depending upon climatic conditions and some other factors that we'll be talking about in a few minutes. But how is decomposition measured? Well, it's generally measured. It can be measured in some different ways, but one of the most common ways that it is measured in a field setting is through the litter bag method. And the litter bag method basically works like this: a fixed amount of litter, and that could be from one single species or maybe a reference litter. It could be from mixed leaf litter as well. It just depends upon the particular study and what is really, what information they're trying to gain. But a fixed amount of litter is placed in some kind of synthetic mesh bag, and it's placed out in the environment. That can be either buried in the leaf litter or submerged if it's in an aquatic habitat. It's put into a mesh bag to allow for decomposers to enter and begin to feed on the litter and break it down. Okay, so the mesh size of the bag is chosen so that decomposers can enter, but it doesn't really allow the plant material to escape. Okay, this is important because after a time, these litter bags are collected and then dried and weighed to determine how much mass was lost over a particular time. So over time, of course, as I just discussed, fungi and bacteria will, and what we're talking about over time is, many months and even sometimes years that these bags are deployed before they get retrieved from the field. Over time, fungi and bacteria colonize the organic material and break it down. And this results, of course, in a loss of mass in the litter, which can be measured. And like I said, it normally takes months for sufficient decomposition to occur. How fast decomposition occurs, though, of course, as I mentioned, it depends on several different factors. One of the important factors that influences how fast litter will decompose is what's called litter quality. Litter quality has to do with the content of the litter, a certain chemical content. So let me just kind of introduce this concept of litter quality through an experiment. So this experiment involved 30 litter bags that were created with 3 different species, litter from 3 different species: sycamore, red maple, and white oak. Each of the bags was filled with 5 grams of litter, okay, and the bags were buried under leaf litter on the forest floor. At 6 different points in time over the course of a year, 5 litter bags of these 30 were collected, and at each time point their contents were dried and then weighed. So what you'll notice is that first of all of the curves there for the 3 different species begin at 100% of the original mass remaining on the y-axis, okay, at the top left point there. And the weight, the dry weight of the litter is shown over time, over a period of 360 days on the x-axis. So what you can see, of course, is that over time mass was lost, and this should make sense to you from my previous description of the litter bag method. Because of decomposition, mass will be lost over time because of the activity of decomposers decomposing that leaf litter material inside the bags. What you'll also notice is that the rates of the mass that was lost over time differed for the three different species. So what could be responsible for this? Well, let me show you some information that might help you come to an understanding. The percentages shown there for each of the 3 different species is the percent of lignin in the leaves. Okay, lignin, you may know, is kind of the complex molecules that form wood. So lignin's a very, very complex chemical compound, mostly derived from, associated with woody material. Okay, and it's an integral part of the secondary cell walls of plants and even some algae. It's one of the most abundant organic polymers on Earth. In fact, cellulose is the other, and is more abundant than lignin. But lignin is a very, very common molecule, and it's present in all in many different plants to different degrees. Excuse me. So the lignin content is often measured in percent by weight, and what you can see is that sycamore has the highest percent lignin content, and followed by white oak and then red maple. And what this the conclusion from this is that the more lignin a leaf litter has, the slower it decomposes. And in fact, this study is consistent with many other studies that have shown the same relationship between lignin content and decomposition rate. It actually gets a little bit more complicated than this, and I'll talk about that. I think it's the next slide. Lignin content can be an indicator of litter quality, but there's actually an interesting twist on this that I'll share with you in the next slide. But at least this data does suggest that lignin plays an important role in influencing how fast leaf litter will decompose. So there's an inverse relationship between decomposition rate and litter quality. So the in addition to litter quality, temperature is also really important, and this probably isn't too surprising. Of course, warmer temperatures, chemical reactions, biological activity all increase, and so it shouldn't be surprising that decomposition rates would be faster under warmer conditions. For example, in tropical areas, decomposition rates are very, very high compared to, say, in the Arctic or boreal or even temperate regions. So this graph, and I'll just go over it kind of quickly here, not to kind of drive this point home too hard, but this graph is basically showing a combination of things. So the first thing that to look at is that on the y-axis is shown rate of mass loss. Okay, so a high rate of mass loss would mean a very high decomposition rate, because the mass is lost at a very high rate, indicating high decomposition, and lower on that axis would be a slower decomposition. On the x-axis, we have two variables shown there. We have lignin content, and we just talked about that as a measure of litter quality, but we also have nitrogen content, and it turns out that nitrogen is also really very important in terms of how fast decomposition rates occur. And it turns out that the ratio of lignin to nitrogen is a much better measure of litter quality than just lignin alone. So this ratio of lignin to nitrogen is commonly used as an indicator of what's called litter quality. Okay. So on the x-axis, what we see is on the left end, where we have low lignin and high nitrogen, we, for the data that's shown here, these are data for different tree species in two different locations, in North Carolina and New Hampshire. North Carolina is warmer in general than New Hampshire, which is at a higher latitude. And so what we see overall between these sets of species that were sampled in these two different locations is that the decomposition rates overall are slower for New Hampshire. Okay, lower rates of mass loss. They occur lower on that y-axis range there. North Carolina, the species the species that are shown there in North Carolina, those are decomposition rates that are in a location that are much warmer. Okay, and so the decomposition rates overall are faster at in North Carolina. What you'll also notice is that the curves decline as you go from the left to the right along that x-axis. And what that is telling us is that the tree species, the leaves that came from tree species with a low lignin content but a high nitrogen content, decomposed at a much higher rate than leaves from tree species that had a relatively high lignin content but low nitrogen content. Okay. So this ratio of lignin to nitrogen is very, very predictive of decomposition rates, and it's been shown in quite a few studies and is a very, very established relationship. So one of the things to take home from this as well, when I talked about sort of the process of decomposition, is that when decomposition rates are high, what this means is that nutrient supply rates are also high. Okay, they're higher at higher rates of decomposition because, of course, the higher rates of decomposition will also result in higher rates of mineralization. That is, the release of mineral forms of nutrients that can be taken up by microorganisms and other plants. Okay, so both temperature and litter quality are important in terms of influencing decomposition rates, and as a consequence, nutrient supply rates. The last concept I want to cover is the role of living organisms in ecosystems in terms of the degree to which nutrients are retained in ecosystems, and we call this nutrient retention. So I'll introduce this kind of through a study. The study that I'm going to introduce was the data was collected by Likens and Borman. They were actually lead researchers in an experiment, a very large scale and long-term set of experiments that were conducted in the Hubbard Brook ecosystem, and those are referred to as the Hubbard Brook ecosystem experiments. And what you can see here is a photograph of a watershed, and a watershed is just simply an area where all of the water drains into a common basin. Okay, and what's shown here is one watershed that has been logged versus a neighboring watershed that was not logged. And what Likens and Borman and others studied was the degree to which the presence or absence of biomass, living organisms in these watersheds, the degree to which their presence or absence influenced the degree to which nutrients were retained or lost from these ecosystems. So as I mentioned, a watershed is an area on the landscape where all of the water that lands in the basin is basically drained from a single location, drains out of a single location. And so in the Hubbard Brook ecosystem study area, which was in New Hampshire, multiple different watersheds were studied. Some of those watersheds, and this was all replicated, a number of watersheds were logged and others were left unlogged. At the bottom of each of those watersheds, a weir was built, and a weir is just simply a construction that funnels water through a finite area such that the water can be regulated, collected, analyzed, and so forth. And so what they did was they collected rainfall runoff in the weirs over time in both the watersheds that were logged and replicate watersheds that were unlogged. And what they did was they analyzed this water that they collected from these watersheds for various different nutrients, particulates, erosional materials, and so forth. And so what they found was that in watersheds that had been deforested, okay, those had lost a lot more particulate matter and therefore also nutrients from the system than the forest watersheds that were not logged. And that can be seen here in the data of total particulate matter that was measured in these weirs in the two different logged versus unlogged watersheds. So the deforested watersheds are shown in red, forested watersheds in blue, and the point at which the watersheds were deforested is shown there in the arrow. And what you can see is that prior to deforestation, total particulate matter that was collected, and this is, by the way, a strong indicator of nutrient loss, because particulate matter That is sort of fine clays and sediments and things that are flowing through these weirs carry a lot of nutrients. And so this actually represents a lot of erosional loss from the watersheds. And it's a pretty dramatic increase in particulate matter that is being lost from the deforested watersheds compared to the forested watersheds. So in deforested watersheds compared to forested, what they found was that overall water loss increased, and this was due to increased surface water runoff. This caused increased erosion in deforested watersheds, and also a dramatic increase in the loss of nutrients, because these erosional materials, soil, silts and clays and so forth, carry a lot of nutrients. And so nutrient losses were observed to increase quite dramatically across quite a range of nutrients, not only just simply the primary nutrients like nitrogen and phosphorus, but a lot of minor nutrients as well from this system. So when we look across different ecosystems, we see that different systems have very different characteristics in terms of where the nutrients are in the system, and this strongly influences the ability of an ecosystem to recover from a disturbance such as like deforestation. In temperate forests, for example, shown here on the left, this pie chart is just showing kind of the distribution of where nutrients are in the system. In a temperate forest, about half the nutrients reside in the soil, and quite a bit in the wood, not quite 50%. The rest is in litter and leaves, litter being sort of the dead organic material, leaves living. In contrast, in tropical rainforests, very little nutrients are retained in the soil, are present in the soil. The vast majority of nutrients are trapped in wood, well over 3/4 as shown here for this particular tropical rainforest, and a small amount in the litter and leaves. What this means is that in these two systems are very, very different in terms of where the nutrients are, and that strongly influences the ability of these systems to recover from a disturbance like logging disturbance. In a temperate forest, because the nutrients are locked in the soil, if the forest is logged, there's still a tremendous amount of nutrients still available in the soil that can allow the forest to recover. So these forests have a much higher nutrient retention, relatively speaking. I mean, any logging is going to cause nutrient losses, but compared to a tropical rainforest, temperate forests have much higher nutrient retention because the nutrients are stored in the soil, versus in a tropical rainforest, the nutrients are primarily stored in wood. And if those forests are deforested, like what we're seeing in the Amazon basin and other tropical areas of the world, and those forests are not replanted or reseeded or managed in ways that facilitate reestablishment of the rainforests, those systems can continue to lose nutrients quite rapidly. And since there's few nutrients in the soil to begin with, those systems recover very much more poorly than temperate rainforests do. So the nature of the biological organisms in any particular ecosystem play a really important role in terms of the degree to which nutrients are retained in that system. Okay, this concludes the lecture on elements in ecosystems.